Hello everyone, I am Dr. A.Vijayalakshmi from Wales Institute of Science, Technology and Advanced Studies, Chennai, Tamil Nadu. In this class, I'll be briefing about the macroscopy, transfer section, and powder microscopy of Ephedra step along with this identification chemical test. Ephedra stem is also known as Ma Huang. It is a Chinese drug. So it consists of dried aerial parts of Ephedra sinica and Ephedra equisetina belonging to the family Nitaceae family. This family is considered transitional between gymnosperms and angiosperms and is as such is very important to taxonomist. And largely it is grown in China, Pakistan and northwest India. Ephedra plant is a small woody dioecious shrub which reaches up to a height of about 60 to 90 centimeter in height. So this Ephedra plant is a small woody shrub which is grayish green in color, which does not have any typical odor, and it is bitter in taste. The stem is thin, cylindrical in shape, much branched, which bears scale leaves at the nodes, and internodes is present. And the leaf is reduced to very small in size, about two teeth sheets, and when the fresh, the herb is aromatic in odor. When it is dried, it becomes odorless, and it has bitter and astringent taste. At the node, it bears a small sized teeth leaves. Nearly two leaves will be present in most of the plants. Sometimes three to four small leaves is also present, and the leaves are decussate and connate. And different species of Ephedra can be distinguished by the thickness of the stem, length of the internodes, number of ridges present, and number of leaves, etc. So usually two species exist. One is Ephedra sinica and Ephedra equisetina. Ephedra sinica reaches to a height of about 30 centimeter of the branches and 3 to 6 centimeter of internodes. And equisetina is more woody, and the branches reaches to a height of about 25 to 200 centimeter with 1 to 2.5 centimeter of internodes. In equisetina, the outer surface is gray to pale green in color and it is smooth in appearance, whereas Seneca, it is brown in color. It is very rough because of the presence of the longitudinal ridges. And in Seneca, a pair of sheathing leaves is present at the node which encircle the stem, and that is fused at the base. And the leaf base is reddish brown in color, and the apex is acute, recurved, and the lamina is white in color. Whereas in Ephedra equisetina, it is brown to purple in color. The lower leaves is black, and the apex is very short and it's not recurved. Coming to the microscopical character, the transfer section of Ephedra shows shows the epidermis, cortex region, pericyclic fibers, vascular bundles, and pith region. The outermost region is epidermis, which is a single layer of thick-walled quadrangular cells present with thick and smooth cuticle. The vertical rows has shrunken stomata, which is present between many vertical ridges of the stem. Below the ridges, we can see a group of non-lignified fibers, which looks like bunches of grapes. And the cortex region is composed of two to three layers of chlorenchyma, which is loosely arranged parenchymatous palisade cells containing chloroplast, and it consists of cortex regions, which is having non-lignified fibers and lignified fibers. The unlignified fibers, it appears like a bunch of grapes, and which is present below the ridges where there is no palisade cells are seen, and lignified fibers are found scattered, isolated, or in groups of two to four. And this is occurs in the inner layer of oval cortical parenchyma, which shows chloroplast. And the lignin fibers consist of pericyclic regions outside the phloem, and about 6 to 10 vascular bundles. It is a collateral conjoined open type of vascular bundles is present. And the xylem is having a well-developed vessels, tracheids, fibers, and parenchyma. And pith region, it is the innermost layer which is composed of a large, thin wall, lignified, and polygonal parenchyma with intercellular spaces. And some cells contains brownish and mucilaginous masses. And the powder of Ephedra stem is pale yellowish brown with faint odor and astringent and slightly bitter in taste. And it contains epidermal cells, which is quadrangular in shape with thick, rigid outer walls and shrunken stomata. And two types of fibers is exist, both lignified and non-lignified fibers with uniform thickness, long and cylindrical, which look like a glass rod. And xylem is present with the bordered pits, and the brownish matter is present in the pith regions. And the chief active constant present in ephedra stem is protoalkaloid, that is ephedrine, and it also contains ephedrine and norepinephrine and pseudoephedrine. And other than this, the other alkaloids which is present is 1-methylephedrine, dimethylephedrine. An identification chemical test to the drug, aqueous extract of the drug, add dilute HCl, copper sulfate solution, and sodium hydroxide solution is added, which turns violet in color. On adding a solvent ether and shaking vigorously, the ether layer turns purple and the aqueous layer becomes blue in color. And the chief use of this drug is it is used in the treatment of asthma. It's a bronchodilator, so it is used in the treatment of asthma, hay fever, rhinitis, and also in whooping cough. Thank you for your patience listening.